Hello, we are Purdue iGEM, a synthetic biology research organization, and we will be presenting our 2021 project, C-ARGO, a rapid detection diagnostic device for COVID-19 that can be adapted to detect any viral pathogen in saliva. Since early 2020, COVID-19 has affected lives on a global scale. Diagnostic tests at the beginning of the pandemic, developed in the form of nasal swabs, saliva tests, and antibody tests, were rushed to the market for mass production, which unfortunately compromised detection accuracy. According to a study done by UC Davis, approximately 2 to 29% of negative early pandemic diagnostic tests were false, meaning that more than a quarter of those who tested negative may have been infected. Further issues were the long and variable runtimes, ranging from a day to 2 weeks. Thus, there was much to be improved within COVID-19 detection. To solve these problems, Purdue iGEM decided to develop a device based on three main principles: reliability, rapid testing, and affordability. To be reliable, we chose an Argonaut-mediated system, proven to be more precise than the CRISPR-Cas9 system. To implement rapid testing, we designed the device using microfluidics to decrease runtime. To be accessible, we focused on using inexpensive materials and equipment to ensure our device could be used around the world. Using these principles, we developed C-ARGO, an accurate, non-invasive, affordable, and fast saliva-based point-of-care diagnostic test system. The concept of C-ARGO works as shown in the following video. First, a sample of patient saliva is input into the microfluidic chip. RNA from saliva is extracted by an embedded chitosan capillary. Next, RT-RPA regions are inserted. RNA is converted to DNA and amplified to produce a large amount of our target sequence. Argonauts and molecular beacons then enter the device. Argonauts cut the DNA to generate fragments of single-stranded DNA. These DNA fragments bind to molecular beacons, causing them to hybridize and produce fluorescence. Finally, this fluorescence is then quantified using a cell phone spectrophotometer to interpret results, which are then output on an app. As shown in the video, C-ARGO includes four main components or biological processes: a chitosan capillary for RNA extraction, RT-RPA to convert RNA to DNA and amplify our target sequence, Argonauts to generate ssDNA of our target sequence, and molecular beacons to identify the single-stranded DNA and produce fluorescence. I'll now pass it on to Grace and Victor to discuss the biologics in depth.
The first component of the system is the chitosan capillary. Chitosan is a bioadhesive polymer that binds either positive or negatively charged molecules depending on the pH of its environment. Since RNA is negatively charged at a broad range of pH values, chitosan-coated capillaries are a perfect medium for affinity chromatography of RNA. The entire process is illustrated in the diagram in this slide. We decided to use chitosan capillaries because of their high RNA extraction efficiency and their rapid action, with speeds as fast as 90 seconds. The capillaries also enable easy test self-administration because they extract RNA within the device itself. The second component of the system is the RT-RPA reaction used to amplify the target DNA. RPA, a novel isothermal amplification method, was chosen for C-ARGO for several reasons. Literature suggests that it is the fastest isothermal amplification system in microfluidic devices, reaching detectable concentrations within 30 minutes. More importantly, its manufacturer sells lyophilized reagents that are stable at a range of temperatures. These features uniquely allow it to be incorporated into a portable point-of-care diagnostic device such as C-ARGO. Furthermore, RPA can be used to produce large amounts of DNA from target RNA through the addition of the enzyme reverse transcriptase.
The third component of the system incorporates Argonaut proteins to cleave target DNA. TTAGO, or Thermus thermophilus argonaut, is a member of a unique class of endonucleases called Argonaut. Argonaut proteins act by interacting with several DNA guides to cleave a specific target region, removing it from that genome. This is illustrated in the shown diagram. We decided to use an Argonaut-mediated system instead of a CRISPR-Cas9 system, as Argonauts are associated with higher accuracy and reduced off-site activity. The fourth component of the system uses molecular beacons to detect target sequences cleaved by Argonaut proteins and to produce fluorescence in response. Molecular beacons consist of several basic parts: a 5' fluorophore quencher, a probe sequence flanked by 2 stems, and a 3' fluorophore. The beacon folds spontaneously as a hairpin structure that quenches fluorescence until the probe hybridizes with the target sequence. This causes a conformational change in the beacon, separating the fluorophore from the quencher and emitting a fluorescent signal that is detectable via a spectrophotometer. Thus, if a significant deviation from background fluorescence is detected, the COVID-19 genome is present in the sample, and the patient is COVID positive.
To design all biological parts of the system, several team members collaborated on a Python script that interfaced with online oligo design tools. This program was used to unify and facilitate the interdependent design processes for RPA primers, Argonaut guides, and molecular beacon molecules. Upon inputting a host genome, the program suggested an optimal Argonaut guide, picked optimal RPA primers to amplify the necessary region, and selected potential molecular beacon sequences to detect the target sequence. The parameters for optimization were found through a well-documented iterative design. Examples of products of the code's optimizations can be found in these images. In 2021, we worked to optimize this program so that it can be used to quickly design a system of compatible biological parts that may be used to detect other viral pathogens with only an input target DNA sequence.
In 2020, we plan to implement our diagnostic assay on a 25 by 25 millimeter microfluidic chip. Since then, we have realized that the use of this microfluidic device would require specific syringe pumps, for fluid flow actuation, which would be costly and difficult to implement at a large scale. Therefore, we switched to a radial design. In it, reagents for each of the steps in our assay are stored in the top disk, and they flow vertically from top to bottom when chambers in multiple disks align. This design allows for compact reagent storage and actuation for simple rotation rather than complex and costly external equipment.
Validating biologics of our diagnostic assay chip began with recombinase polymerase amplification process along with the optimal primers designed by ourselves. We ran an agarose gel containing a positive control, a negative control, and a reaction with optimal primers. We were able to successfully confirm amplification of the target viral sequence by confirming a thick, amplified lane of expected size. Following the RPA was the molecular cloning of 2 Argonaut proteins of interest. As mentioned previously, we chose Thermus thermophilus argonate, or TTAGO, and a previously cataloged part, or H2AGO, as a gene of interest, and attempted to clone into the backbone over the summer of 2021. We faced difficulties, however. White-6 sequencing revealed the incorrect transformation of both constructs multiple times. In order to troubleshoot seeing no transformants, we attempted to replace the codon-optimized Argonaut insert with non-codon-optimized inserts, which was proven to be a successful ligation to validate cloning protocols and procedures taken. Additionally, fresh reagents and high-fidelity double digest restriction enzymes were ordered. Upon troubleshooting, we were able to successfully clone the codon-optimized sequences of both Argonaut proteins into DH5 alpha and BL21 strains of E. coli to further continue the protein expression process. I'll be passing on to Haley to talk about further progresses about protein expression and purification.
During the expression and purification process for both proteins, several problems arose. 2 induction methods were used to induce protein expression. Upon induction, various purification methods were tested, including sonication, chemical lysis, ultracentrifugation, and column purification. SDS page gels were run to validate expression and purity. The gel results showed insufficient purification of the protein. The gel also demonstrated a slight difference between the uninduced and induced samples, indicating some protein expression. However, expression was very low, and a more quantitative method was devised to verify expression. Due to time, additional quantification and experimentation on purification and production of the protein could not be conducted. In conjunction with the molecular cloning, COVID-19 specific phosphorylated guide DNAs are designed to initiate the first round of TTAGO cleavage in the device mechanism. The efficacy of the guides and their ability to cleave with specificity was tested using TTIAGO from NEB. A cleavage assay was run and the products were run on a urea page gel. The gel showed successful and specific targeted cleavage, demonstrating successful design and implementation of the three-guide DNA cleavage system. The picture shown to the left was a cleavage assay carried out with 1 UL of guides, and the one on the right is with an increased amount of guides. Here, we see that the targeted sequence, which will later bind to our molecular beacon to ensure detection. The final part of our biological testing assay involved the use of molecular beacons for quantifiable fluorescent detection of our target sequence. A cleavage and limit detection assay was designed to test the fluorescent capabilities and readout accuracy. However, due to time constraints, this experiment was unable to be conducted, and we do not have results to share with you today.
We were able to characterize the RNA extraction process using a 5 by 5 millimeter glass fiber pad coated with chitosan. We used a Langmuir isoform absorption model to determine maximum capacity equilibrium constant of our system for varying RNA concentrations. This model relies on the assumption that only one layer of RNA combined to chitosan, there are no interactions between adjacent RNA molecules, and that adsorption-desorption is an equilibrium reaction. We validated our model experimentally and estimated binding capacity of approximately 6,900 nanograms. This value is above those reported in literature for other platforms and above the limit of detection for our downstream reactions. However, at lower concentrations, the amount of bound RNA decreases considerably, which could be a problem for patients with low viral loads. We also wanted to test the versatility of our system by varying the sample and elution volume for a fixed amount of RNA. At higher sample volumes, the amount of RNA recovered decreases considerably due to the initial dilution of the sample. Whereas for varying elution volumes, for volumes over 100 microliters, the amount of RNA eluted did not increase significantly. Therefore, we believe our system is capable of operating within the range of 50 to 100 microliters, which is typical volumes used for our downstream reactions as well.
In 2020, we talked to professionals who are involved in the testing and management of microfluidic devices here at Purdue. From these discussions, we learned how to optimize their device, manage mixing and actuation, and learn about the diagnostic device requirements upheld by the FDA. Dr. Worley helped us optimize their device by removing dead space. Dr. Linus helped us optimize their device by utilizing vibrational mixing and also adding an extra valve to account for pressure differential. Dr. Gates is a former regulatory affairs personnel from Pfizer who gave us valuable insight on the FDA requirements and the importance of regulation. In 2021, we talked to Dr. Esteban Ramirez, the Chief Medical Officer for Protect Purdue, Purdue University's COVID-19 spread prevention campaign. We also talked to Dr. Rebecca Wilkes, an associate professor and molecular diagnostics section head for molecular and virology within the Department of Veterinary Medicine at Purdue. From both of these interviews, we were able to gain valuable insight on how to improve our device for large-scale implementation, such as the surveillance testing being done here at Purdue.
While it may seem that our device loses relevance with the introduction of COVID-19 vaccines, the nucleic acid-based molecular diagnostic assay can be applied to detect a characteristic sequence of any viral pathogen. The program we previously wrote to design the biological parts used to detect SARS-CoV-2 can be used to produce multiple sets of compatible RPA primers, Argonaut guide sequences, and molecular beacon sequences, given a characteristic sequence of a viral pathogen. We hope for future researchers to employ this system to help facilitate the fast design and implementation of diagnostics for future viral outbreaks. Ultimately, we hope to show that C-ARGO is more than just a COVID-19 test. It is the future of diagnostic devices. Thank you all for listening and have a great day.